Okay, so today GoINPHARMA meets Dr. Nello Mainolfi, founder and CTO of Kymera Therapeutics Limited. Nello is the founder and CTO of Kymera Therapeutics, the Cambridge, Massachusetts-based biotech company pioneering targeted protein degradation as a transformative new approach to creating breakthrough medicines. Nello, born in Napoli, Italy, has received his PhD in chemistry from Imperial College, University of London, and has continued postdoctoral training at the Scripps Research Institute in California. He then started his professional career at the Novartis Institute for Biomedical Research in Cambridge, Massachusetts, where he advanced several programs from research into preclinical and clinical development. While at Novartis, he also championed new technologies such as using fragment-based drug discovery as a core strategy to deliver multiple development candidates. Nello is also entrepreneur and resident at Atlas Venture, a Cambridge-based VC firm focused on investing in early-stage biotech. Nello is married with Paola Castaldi, also a scientist manager at AZ and father of two. He enjoys running and sailing in his spare time. So thank you, Nello, for sitting down with us to talk about your company today and the idea behind it. How did it begin, and what has been the journey so far?
Well, thanks for the opportunity to really contribute to these conversations around startup and around innovating science. So the journey really started towards the end of 2015, and it came as a really unique opportunity where there was a combining event of the field of targeted protein degradation that has been for many years relegated to really a proof of concept in academic labs, so the field was maturing, and we saw an opportunity to take this really cool technology and apply to drug discovery, to real drug discovery. And the other aspect was really the potential behind the technology. We were able to see, and we were not the only one, but clearly one of the ones that decided to invest in this. We were able to see that the power of the technology was really to go beyond the the so-called druggable proteome. The transformational promise was to really go after proteins that we know are disease causing, but didn't really have a technology that was able to target them in a therapeutic way. And so it was really at the end of 2015, early 2016, where again, as you mentioned before, also at my role with Atlas Venture, one of the partners there, Bruce Booth, and I really sat together and thought that the time was really right to take the next step and invest into an investment thesis that was around taking this early technology, making it ready for therapeutic modality, and really trying to develop innovative therapies for patients. And so the initial investment was small. It was around $3 million to really prove the concept that the path that we were taking would be fruitful. And so we generated really in the first 6 to 9 months really lots of data, some around our ability to therapeutically target traditionally undruggable proteins, but also, and I think this is what really separates Kymera from the other companies that are venturing into this space, we were able to generate data that elucidated more closely and more deeply some mechanisms around targeted protein degradation as we know it today. And so this is really what allowed us in a really short time to generate enough interest and excitement in the investment as well as in the pharma industry that allowed us to then close our Series A in mid-2016 and to really fund the company to take it to the next step.
Okay.
So just maybe to add a couple more points about what happened also in the early stage. We've been aside from building the science and building the investment, continuing to build the investment thesis, really the main job was building the right team of people that believed in the technology and also believed in wanting to generate new drugs to tackle unmet medical need. And so we found so far a group of 18 team members that work in Cambridge in our headquarters, and we have a network of collaborators around the world that really transform our headcount from a small biotech, as I said, of 18 to more than 50 and 60 people around the world.
Wow. Wow, that sounds amazing. You touched on a few points already, but one of the most innovative aspects of your company is the way you go about drug discovery. So can you go into a bit more detail on how you implement it and what are the benefits of the approach compared to more conventional ones?
Yes, thank you. So it's in a way, at the really high level, it's quite a simple concept. Although once you go deeper into the concept, you'll see that there are some nuances and features that are quite complicated. But let's stay at the high level for this interaction today. So we start with the premise that traditional small molecules that are used to inhibit the traditional drugs that we use almost every day are made of small molecules that inhibit a particular function of, let's say, a protein, a receptor, and an enzyme. And that inhibition per se is able to generate a phenotype that can ameliorate or cure a given disease. The problem is that historically only about 10 to 20, some people say 15% of proteins in our cells are targetable through this technology, through inhibition of a particular function. And so basically all the work that has been done in the past 20 years or so in understanding and refining and mapping our genome and hence our proteome hasn't been really captured because we have an inability to target proteins that don't have this catalytic function. And one of the key really promise of this technology is the ability to go after those proteins. So the other 85% that don't have a small molecule that is really driving or a substrate or the catalysis of a function that can be inhibited by a small molecule. And so we basically, the way that our technology works is we use I don't know if we're seeing the graphics that we've provided, but we use these heterobifunctional molecules that are showed in purple on the graph that are able to work through affinity. So we're not inhibiting any function. We're basically binding to two proteins. The protein on the left in blue is an E3 ligase. The protein on the right in green is a target protein of interest. Our molecules, almost like allowing a kiss of death, are able to bring these two proteins together. And the E3 ligase, which is part of our innate protein recycling machinery, tags the protein with a chain of ubiquitins. This polyubiquitin process tags the protein of interest for proteasomal recognition. So once the proteasome recognizes this artificially now tagged protein, the signal is that the protein needs to be destroyed. And so this causes the destruction of the protein. And so really the transformational ability here is to go into a cell system that has a malfunction that is causing a disease. And we're able to go there and bind to this protein. And by, again, utilizing our protein machinery, destroy that protein. And with the ability then to remove this disease-causing mechanism.
Okay.
And maybe just to add one more sentence here at the end, really, and this is why there is so much interest really globally around this technology, because it allows you to do things that you could not do before. And there is a lot of hype around CRISPR technology that I'm sure many of your readers are aware of. And this is really, you can view this technology as a small molecule version of that CRISPR technology.
Yeah. So the very core of your technology is the regulation of cellular processes via redirecting the ubiquitin proteasome system, UPS. What is the future there? Do you have any clinical trial planned in the near future?
Yeah, thanks for the question. So for sure, our plan is to become a clinical stage and eventually a development stage and commercial stage company. So for the most advanced programs, we plan to be in first in human around 2020. And this is why we've been funded so far, and we continue to be funded in order to really prove that concept in the clinic and show that this powerful technology can actually impact unmet medical needs in the clinic.
Wow, that's not too far away. So in October last year, Kymera Therapeutics has been able to raise 30 million in a Series A financing. You mentioned that. But what do you think that the important investors such as Atlas Venture, Lilly Ventures, and Amgen Ventures really convinced? What convinced them to go for the financing?
Sure, yeah, and as I mentioned before, I think hopefully is also clear to your viewer and readers that the potentials here are huge. The ability to go after disease-causing proteins that before we could not go after is really an opportunity that is almost impossible to pass on. And so I think that was really what catalyzed the conversation. And I believe on top of that, which I think differentiated us from the field of drug discovery in general, is the fact that Kymera was able to generate really enticing data on both the therapeutic programs, but also on our ability to go into the depth of this technology and understand the kinetics, the substrate specificity, the type of E3 ligases that one wants to use with the particular target of interest. And so this is really the investment thesis around Kymera, and this is what really makes us a unique company right now.
Yeah, let's see, yeah. And talking about the future, what's next for you and Kymera?
Yeah, so as you might imagine, small companies are always full of activities, and so every year there is lots of goals on the science, on the corporate, on the financing. So for us, after 2017 was really the year in which we propelled a really small startup into a more early biotech phase. 2018 is really for us to take both the team and the company to the next step. So we plan to grow the team to almost twice the size that we have today, and more importantly, we are continuing to talk with either potential pharma partners as well as in the investment community to continue to fund the company in a way that would allow us to go into the clinic as soon as possible. And as we mentioned, 2020 is our goal. So we will have actually some really exciting developments that will be announced in the next few weeks and months about inroads that we made in all those activities.
Okay, well, thank you. It sounds like we need to interview you again in the very near future.
Sure. Really thanks for the opportunity to share some of the story, and looking forward to more interactions.
And it sounds really exciting, and all the best from us.
Thank you.